Okay, well, welcome to the November. It's hard to believe it's November already. Artificial Intelligence for Clinicians, the AI4C lecture series, presented by our Mass General Brigham AI. And today we are thrilled to have Dr. Andrey Fedorov, who is going to talk about the imaging data commons and work he's done there with data, tools, compute to power your AI explorations. And he's from Brigham and Women's Hospital and Harvard Medical School. As you know, this is a series that we do every second Thursday of the month. And we go from September to May. We have a great lineup. This is our fall speakers. If you missed it, you can get them on the recordings, which Carrie Ann has put that link in the chat. We had Adam Flanders from Thomas Jefferson and also the board of the RSNA. We had Will Su from UCLA. Today we have Andrey, and then we have Safwan Halabi from Northwestern of the Children's Hospital. And he's doing a lot of work in AI on the pediatric patient population. So that'll be great. And then we have a great winter and spring lineup as well. But again, please go to the link there if you want to see any recorded past lectures over the past number of years. And you can also look for us on LinkedIn and Twitter, follow what we're doing on Twitter and so forth. So today, as I mentioned, we are delighted to have Dr. Andrey Fedorov, who received his Bachelor of Science in Information Technologies and Engineering from Ternopil Academy of National Economy in Ukraine. Let's see, I have that wrong. Ukrainian National University in Ukraine. I'm sorry, Andrey, for that, but he's from the wonderful country of Ukraine. And he got his PhD in computer science from the College of William and Mary in Virginia. He actually did some summer internships in the surgical planning lab at Brigham and Women's. And then after he finished his PhD, he joined the lab, and he's been here for 15 years. It's been wonderful to have him at the surgical planning lab in the Department of Radiology at Brigham and Women's Hospital. And he has worked on translation and evaluation of image computing tools in clinical research applications. And he's focused on quantitative imaging, imaging informatics, and image-guided interventional procedures. He's done a lot of work with that. He's currently a principal researcher at Brigham and Women's Hospital and associate professor of radiology, Harvard Medical School. He's been really devoted to advancing the role of reproducible science and data sharing and open source software for academic research and developing infrastructure and tools for best practices to help researchers improve transparency of their studies and make their analyses more easily accessible to others. He's received funding from the National Institutes of Health, National Cancer Institute, and he's contributed to a boatload of open source projects, most notably 3D Slicer, which I'm sure many of you have used or are using. And if you haven't, you should check that out. Together with Ron Kinkinis, who is also at Brigham and Women's Hospital and part of the SPL founding member, he served as a co-PI of the Quantitative Imaging Informatics for Cancer Research Project that focused on developing, again, open source informatics technology in support of quantitative imaging biomarker development and interoperable sharing of image biomarker data using the DICOM, the Digital Imaging and Communications in Medicine Standard. Again, together with Dr. Kinkinis, he's currently co-PI of the consortium that's tasked with building the National Cancer Institute Imaging Data Commons. And that's what he's going to share with us today. And if you're going to RSNA, he runs a wonderful hands-on workshop that will show you, you'll get to use some of these tools. But today he's going to talk to us again about the Imaging Data Commons, data and tools and compute to power your AI explorations. So Andrey, let me stop sharing and you take it away.
Thank you so much for this very kind introduction, Kathy, and for inviting me to present for this meeting. It's a big honor and pleasure. So let me share my screen. Just wait. Can you see? Can you see it? Okay, excellent. So thank you. So yes, so today I will talk about Imaging Data Commons. It's not the first talk I give about this project, but I restructured it a bit differently, in part to address perhaps the interest of this audience, which is more clinical, but I hope very interested in AI as everyone these days. So I will start with the disclosures. This project is funded from federal funds. I do not represent here the views of NIH or NCI. I speak on behalf of myself. We work very closely with commercial cloud providers, specifically Google and Amazon, but we do not have any financial relationships. We are working as partners in enabling sharing of public data with the community. And also, we are very grateful to the Access project. I will briefly talk about it, supported by National Science Foundation for providing the cloud compute resources. So Kathy very nicely gave a little bit out about my background, but I wanted to start this presentation with just kind of going back to 2005 when I first came to the lab, and that's how I was introduced to medical imaging, because that experience defined to a large degree my career and what I'm doing now. I definitely, if not that internship, I would not be here today. So back in those days, there was a lot of effort into development of technology to support estimation of brain shift during open brain surgeries at BWH in the open scanner. And back in those days, I contributed to some of the tools that helped with non-rigid registration. And here on the right, you can see kind of the high-level overview of this setup that we put together at that time, where imaging was done at Brigham. We transferred the data to the supercomputing facility, did all the computation there because it was not possible to do it on premises, and then sent it back while the surgery was still going on. We did it on a handful of cases, but we demonstrated the feasibility and utility of this high-performance computing technology. And but more importantly for me, I think not so important were the papers that we published, but my exposure to open source technology, because these were very exciting, very early days of open source, where such projects as Visualization Toolkit, Insight Toolkit, and 3D Slicer were relatively early in the development. And I never worked in open source before. And to me, it was such a empowering experience, because without any prior experience developing medical imaging tools, there is no way I could be able to accomplish anything. Also, back in those days, there were very few publicly available data sets. And I think back in those days already, a surgical planning lab led by Ron Kikinis was at the forefront of sharing data at very small scale. I mean, this data set that we used back in those days contained just 10 samples of the tumors. But back in those days, that was a lot of data. And without this public data set, again, this research would not be possible. So that's what was happening back in 2005. And if we look where we are today as a community, a lot of things changed. And it's really incredible just to see this contrast, because now if you do not see open source implementation of the next greatest AI tool, something is wrong. Most of the tools that you will find in the academic literature now, it is expected that they would be accompanied by open source implementation. Also, thanks to the efforts of National Cancer Institute and specifically the Cancer Imaging Archive, there is explosion in availability of imaging data across domains, including annotations, accompanied by rich and multi-omics data to really empower the research. And more lately, now there's also increasingly there are requirements for funded federally to share data. And finally, there is, you know, we all experience this explosion in capabilities from GPUs, computational platforms. But it's not just about the increasing power and capabilities of those tools, but also access to those tools became a lot easier. So now, in our days, everyone, literally everyone, can relatively easily access virtualized resources that contain GPU, and you can get started with experimenting with AI applied to medical imaging in no time. So with all those opportunities come also challenges. Technology develops very quickly. It can be difficult to understand and put things together. And especially when you are a newcomer in the field, it can be really mind-boggling. So at the same time, with all this technology, especially with virtualization, we can try to change how things are done. Instead of doing things like we've been doing it back in 2005, where we would use on-premises computer resources, set up tools, download data, and do the analysis. Now it's possible to virtualize this as a cloud-based environment where tools, data, and computational resources are located in one place. And this makes it easier to manage data, to make research more reproducible, to share data and tools with your collaborators, and help streamline and advance this whole environment. So from this concept of cloud-based computing or data commons, emerged the National Cancer Imaging Institute Imaging Data Commons. So IDC is a cloud-based environment that contains publicly available data collocated with the various tools that are designed to help you easier use this data, and also resources that can be used for analysis of this data. And importantly, IDC is a component of a broader cancer data ecosystem developed by NCI, which includes repositories of different types of data, genomics, proteomics, canine clinical trial, along with the various tools that simplify access to the compute resources and integration of data from one domain with other types of data. So we received funding in 2019 to work on establishing imaging data commons, and this project has been funded since then, with me and Ron Kikinis being the co-PIs of the project. So the way I want to present IDC to you today is think about it as three components. First of all, it's of course data. Imaging data commons is about data. But then also what kind of tools we give you to help work with the data. What is the added value that IDC brings? And also how we make it easier, hopefully, for you to leverage the computational resources. So as of today, we have more than 78 terabytes of publicly available imaging data. This includes radiology and digital pathology. These are the primary domains of data that we have. And at this point, we probably have about 50-50 between radiology and digital pathology. This covers clinical, preclinical, canine, phantom imaging. But along with the imaging data, we also increasingly have more and more image-derived data. So data becomes a lot more accessible and a lot more usable when it is accompanied by segmentations, annotations, radiomics features. And we see the role of imaging data commons in continuously increasing the richness and the value of data. And finally, even though clinical data is not imaging data, but very often in order to use imaging data in a meaningful way, you need to know more about the patients. And that's why we collocate clinical data along with the images and help you navigate this data. IDC ingests identified images from various sources. We are primarily working with large data collection initiatives and existing data curation centers. We started with the Cancer Imaging Archive as the most established repository that exists to ingest publicly available data from TCIA. But then increasingly over time, we are adding more and more resources and various programs based on the priorities established by the National Cancer Institute to bring more data from a variety of sources to our users. However, ingestion of data into IDC is not just like copying this data and making it available for download. We do a lot of work to harmonize the data, to ingest it in such a way that you can visualize the data, you can search the data. It's represented uniformly so that it's easier to use it with your tools. And we design what we hope should become best practices, how to use various existing tools and cloud resources for cancer imaging research. Beyond images, as I mentioned, we share analysis results and annotations. Some of those we ingest from other sources. Some of those are contributed by users and dedicated projects that focused on annotations. And some of those we, as a project, generate ourselves. When we identify promising analysis tools that are robust and can effectively enrich our data, we would apply those tools to IDC data and share the results with the community. This is a high-level view on various aspects of data that we have right now. So as you can see, we have a variety of modalities, CT, MR, annotations, segmentations, artifacts available for those images. And these cover different organs in the body, different cancer types, a variety of manufacturers and vendors. And a large component of our data is the annotations. So what we hope with this repository is to have a publicly available resource that you can use to design new AI algorithm, that you can use to evaluate existing AI algorithm, that you can use perhaps as a source of teaching materials if you're a radiologist, or test your products or new tools if you're a commercial vendor, because most of the data that we have, absolute majority of data, does not restrict commercial use. So next, I want to go over some example sources of data, because these highlight a variety of data that comes into Imaging Data Commons, and also highlights the added value that we bring by ingesting the data into IDC. So I hope that most of you are familiar with the Cancer Imaging Archive. IDC and TCA are partners in bringing you the high quality imaging data. At this point, we have regular meetings. We work very closely on data ingestion from TCA into IDC, and we have most of the publicly available collections from TCA available also in Imaging Data Commons. But here is one example of how we improve the value and bring added value by ingesting data from TCA, in this case, into Imaging Data Commons. So perhaps some of you may be familiar with the National Lung Screening Trial collection. It's one of the largest collections available in TCIA. It has about 26,000 patients, more than 10 terabytes of data. And since recently, it's a publicly available collection. It's no longer a limited access collection. But if you go to the Cancer Imaging Archive, you will see, and these are just facts, that if you want to search radiology imaging data, you have one portal. For pathology, you have a different portal. You cannot combine this information. You cannot combine clinical data to build cohorts of the images and cases. And the visualization capabilities provided by the Cancer Imaging Archive are, at this point, quite limited. If you look at what we have for the NLST collection in IDC, we have single portal where you can side by side search pathology and radiology. We have viewers that will show you not just the images, but also annotations available for radiology and pathology images. You can search clinical data, metadata for pathology and radiology data using the same interfaces. And also, most of the images for NLST that you will find in Imaging Data Commons are accompanied by volumetric segmentations produced by Total Segmentator, and also by radiomics features. So you can build cohorts not just based on the clinical characteristics of the patients, but also on the characteristics of the structures that are segmented and available in these images. And you can combine all of this information to build very powerful cohorts. And we have tutorials and documentation how to do this. Another data source, Visible Human project. So this project also was probably one of the highlights back in 2005, because this is the data set that drove the development of Insight Toolkit and many other image analysis algorithms back in the days. And despite the fact that this data set is quite old, it's still quite unique, because it's very difficult to find similar data sets where you have the entire human body scanned using CT and MR, and at the same time accompanied by digitized cryosections. And you can really develop better understanding of how those images relate to each other and to the appearance of tissue in visible photography. So this data set definitely has been available for a very long time. But if you go to the National Library of Medicine website with this data set, and you take a look how you can access it, you will have this kind of interface where you have to browse the directories on the website, download one slice at a time. You will have to deal with very strange formats which are encoded in vendor-specific proprietary representation. So, and you will not find any visualization tools available to you in the NLM website to better understand those images. We ingested this data set about a year ago into Imaging Data Commons to make it more accessible. You can visualize all of those images using our portal. You can download either the entire collection or specific series. You can search it. And it's a similar example how you can do more with the images that are ingested into IDC. So it's not just about creating another place where you can download them. It's empowering you to do something new with the data. So the previous two sources I discussed, TCIA and Visible Human Dataset, the actual data can be downloaded from other places, perhaps in a bit different representation, but it is available. Unlike those sources, Childhood Cancer Data Initiative is collecting data which is not available, imaging data, which is not available anywhere else other than Imaging Data Commons. So CCDI is a relatively new initiative which is focused on improving our understanding of child cancers, children and young adult cancers. And this program is very active. There is ongoing collection of data, both radiology and slide microscopy. And sharing of data is one of the big pushes of this program. We are partnering with CCDI to ingest the data from this program, harmonize it, and make it publicly available, searchable, viewable through the infrastructure that is available within Imaging Data Commons. At this point, we ingested about 4 terabytes of slide microscopy data from CCDI. You can see it's quite a range of cancers and anatomies. And very importantly, the images that you will find in IDC are also linked with the other relevant data that is available from CCDI Hub. So through the identifiers that we include in the images, you can find this accompanying data. You can learn more about the participants. You can learn more about the phenotype of this disease, and hopefully build the models that will help us treat those cancers. So hopefully with this brief, very brief overview of some of the sources of data, you understand better how we are different from other archives and repositories. And the difference really is in the name. It's archive versus the commons. So the commons is a resource which is shared by all members of the society, and all of the members of society can access the resource and work together to improve it and to nurture and to grow it. On the other hand, archive purpose is to ingest the data, make sure that it's stored persistently, and make sure that it's accessible to everyone. So this is the fundamental difference. We partner with TCI as a resource that ingests, de-identifies, and distributes the data, but then we are working to add these additional capabilities through the infrastructure and tools, examples, and best practices that we are working on. There are some rules that apply to all of our data for every single image, every single collection we have in IDC. It's publicly available. You can search, download, visualize it without the need to log in, no forms to fill out. And all of the data is available from cloud buckets. So it's available through standard S3 API, which means that you can download it very quickly. It's replicated redundantly using the commercially available resources from Amazon and Google. And we mirror our data between these two providers. We do not forbid commercial reuse of data, but you have to acknowledge the contributors and repository if you use this data in your research or commercial development. Unlike other sources where you can find data, IDC data is versioned. So if for any reason the files are changing, you will still be able to access the previous iteration of the collection, unless some of the data had to be retracted due to violation of PHI, identification of patient data in the images, which is a very rare event. And finally, all of our data, both images and image-derived data, is harmonized into a common format, into DICOM. And this decision is really critical to the implementation of the capabilities that we provide to the users. So in radiology domain, DICOM standard is absolutely ubiquitous. Pretty much every single scanner you will find will generate images in a DICOM format. In digital pathology, it's a bit different. While there is a push towards adoption of DICOM in digital pathology, and DICOM does have the capability to meet the needs of this field, most of the vendors do not produce images in DICOM. And that's why for those images, we develop tools how to harmonize these vendor-specific representations into common DICOM format. And finally, when we talk about image-derived data, such as segmentations or radiomics features, of course, most commonly, those are not represented in DICOM representation, but they can be represented in DICOM. And unifying all of this data into the common format opens a lot of opportunities. DICOM is implemented by a variety of both open source and commercial tools. Once our data is in DICOM, we can use off-the-shelf components from Google Healthcare to provide data hosting. We can use at the same time open source tools to enable visualization. And it's an industry standard. We do not want to introduce any specific IDC-specific conventions. We want to leverage all the work that went into development in our community. And of course, by utilizing DICOM, we ensure that the metadata that accompanies our images, annotations, follows the same data model, which makes the data searchable, which makes it possible to reference the data through the consistently maintained unique identifiers, and which also enables us to be able to accommodate new types of data under the same data model in the future. So DICOM is a growing standard, it's a dynamic standard, it can be improved, and we are also refining the standard based on the needs of our community and data. While we use DICOM standard internally, we definitely do not expect that every single tool will implement DICOM support and will work with DICOM natively. Because of this, we are also establishing best practices how to go from DICOM to the tool-specific representations. And fortunately, in many cases, this process is straightforward. Where there are gaps, we are actively developing tools to make this transition smoother and feasible. This is a high-level overview of kind of the data flow and how we make the results available to the user. So we deal with a variety of data sources from where we ingest. These cover different types of data. This data, if necessary, is harmonized into common representation and undergoes our ingestion processes. And once the data is ingested and released in IDC, the files become available for download through S3 API from AWS and GCP buckets. We ingest the data into Google Healthcare DICOM store, making data available through DICOM web interface. We extract all of the DICOM metadata and make it searchable through, again, standard SQL queries. And for a subset of this metadata, we share it through the Python package that we maintain. But these interfaces really are the underpinning of various tools and capabilities that we provide in viewers, search portal, dashboards, and so forth that I will discuss next. So this is my second part of the presentation, where I will go briefly over the various tools and capabilities that we are developing. And as a high-level overview of those categories of tools, the first category are the tools that really make it easier for you to search and access the data available in IDC. Those tools we develop ourselves. Those tools rely, in some cases, on the standard interfaces implemented in commercial tools. And these also include both tools that we want to be usable for non-technical users, where you would interact with the application or a website, but also enable technical users who can do advanced searching to be more kind of granular in building cohorts and accessing the data. So this includes both interactive tools and programmatic interfaces. Next set of tools is the analysis tools. And in IDC, we do not focus our efforts on developing new algorithms, new AI models. But what we want to make sure is possible is ease of applying those existing open source tools and models to the data in IDC. So we work diligently to make sure that even though our data is in DICOM, you can use those tools. And you also understand how availability of computer resources, uniform representation of data makes the analysis more robust and more rigorous. Finally, we also develop examples how to use existing tools with IDC. So that's all about demonstrations, notebooks, and reproducible workflows. Those we are also contributing. So the entry level to Imaging Data Commons is our portal. That's our kind of first tool that you will encounter. So this is a website where you do not need to log in. You have on the left-hand side, you have the characteristics, a small subset of metadata describing what we have in IDC. You can filter the data by selecting those attributes. And then on the right, you will see collections. From collections, you can go down to the individual patients that are included in the collection, studies. And then from there, you can also open the selected study in the in-browser viewer. So no installation is needed. You do not need to download anything or install anything or log in. So this is our portal. We have, I believe, very powerful visualization capabilities. IDC portal integrates Open Health Imaging Foundation Viewer version 3 and version 2. And this is a very active, very strong and currently supported project, which is funded both by federal grants but also by commercial entities. It's fully open source. You can deploy your own instance of the viewer, and we work very closely with the OHAF team, not just to use this viewer, but also actively contribute the missing features to this viewer based on the peculiarities of the data and the needs of the cancer imaging community. In addition to OHAF viewer, we also have integration with the WallView viewer provided by Kitware. And we are also, the way data is structured, we are open to integrating new viewers based on the needs and constraints that we have. So in a similar way, for visualizing microscopy, slide microscopy, digital pathology images, we provide integration with open source Slim Viewer. Slim Viewer shares some of the underlying components with OHIF, but it customizes the interface to better meet the needs of the microscopy community. This viewer was developed by Marcus Herman, who was here at MGB, MGH. Unfortunately, Marcus left, and he's now working for industry, but his legacy lives on. And since Marcus left, we have been maintaining this viewer, adding features and fixing bugs to make sure that it meets the demands of Imaging Data Commons and our users. OHIF viewer, a Slim viewer similar to OHIF has the capabilities to view not just the images, but also annotations. It supports various types of annotation that you can visualize, both these high-level outlines of the regions of the slide, but also very granular annotations, for example, of individual nuclei or pixel-level segmentations or heat maps that you can overlay on the slides. Right now, I'm highlighting here just these large region-level annotations, and in this case, it's one of the collections from the CCDI initiative. But we are actively working, and we already have some data sets in the pipeline that will highlight other capabilities of Slim in visualizing other types of annotations that will be available in IDC. So one of the pieces of functionality we've been continuously refining over the past 5 years is how to make it easier for our users to download the images that they identified. And you won't believe how many iterations how many ideas we had. But I think right now we are in a good place where we hope that it's relatively intuitive, because our idea is to provide different means of downloading data from various interfaces, because we never know how the user will become interested in accessing specific subset of IDC data. So first of all, you can download what you find in the portal. As you browse through the content in the portal website, you will see this little button. If you click this button, you will see the instructions how to download the corresponding files. You need to do this once. You need to install this prerequisite. But after that, downloading individual collection, study, or series is a one-line command in the terminal. If you happen to find the images you are interested in the viewer, you can do in a similar manner. You can push this button in Archive Viewer or this button in Slim Viewer, and you can download what you see on your screen and use these images on your computer. We also added the capability to 3D Slicer. We established a new extension that allows you to browse the content of IDC and download individual collections or individual cases. You can also copy the identifiers of patients' collections studies into Slicer IDC browser and download those and visualize them in 3D Slicer and experiment with the tools, analysis tools available in 3D Slicer. So this tool is probably easiest to use for clinicians who may not have Python or who may not know how to install Python or who do not want to deal with the terminal. With 3D Slicer, it's all point and click. You download the binary for 3D Slicer, you install extension, and you're ready to go. And if you want to learn more, I have the links here to the forum post that explains in a bit more detail how you can set this up. And I will share the slides after the meeting. I will have the link in the last slide that I showed. So don't worry taking snapshots and so forth. So for the technically savvy users, we developed IDC Index. It's an open source Python package. If you want to write a code that filters data from IDC and downloads, it's this simple to get started. We also have the command line tools that allow you to download content defined by the manifest that you can build using IDC portal or by referencing the unique identifiers for the patient's collections studies. But IDC Index is a lot more than just downloading the data. It has very powerful capabilities to search the data, to build cohorts, to generate links that you can open in the viewer to visualize the images. To generate citations for the cohorts that you build. It's a package which we introduced this year, and we have a lot of plans introducing more functionality into it. Would be very happy to hear feedback from you if you see something is missing or something is not working. And we have a basic tutorial how to get started here. This slide is a bit overloaded, but I wanted to have it here so for a reference, so to speak, because if you ever worked with an LST collection available from TCIA, you know that you would have to download various CSV files. You need to parse them. You need to map the codes to human readable terms. So it's a lot of work before you can do queries and before you can combine clinical data with the images. With IDC Index, we are trying to make it easier for you. So what is happening in this block, after you install IDC Index and instantiated IDC client, you can fetch all of the clinical data available for IDC collections. You can, with just one line, filter all of the clinical data attributes that are available for the NLST collection. And you can list what are those attributes that are available. And then you can access the actual clinical tables. Clinical data tables, which you can link with the images by using this DICOM patient ID. So with just a few lines of code, you take care of all of the searching, downloading CSV files, parsing them, understanding how they're organized, reading documentation. So this is one example where the tools that we provide hopefully make it much easier to accomplish tasks that are difficult otherwise. And we have a tutorial about this. It's linked. It's linked. I forgot the link, but yes, we have a tutorial in our tutorials GitHub repository, which I will share with you later. So IDC Index is kind of entry-level tool for technically savvy users, but if you want to dig deeper, we also provide searchable access for all of the metadata attributes available in our IDC data. So this is really more than 1,000 different attributes. And with this scale of data, thousands of columns, many millions of rows, and very complex, sometimes deeply nested structure of those attributes, you need very powerful backend to be able to efficiently query this data and build the cohorts. So for that, we rely on Google BigQuery product. and it provides a standard query language SQL interface for navigating this metadata. And as an example, this is one kind of query that just to give you an idea how it looks. And in this query, we select all of the slides that were scanned at 40x magnification for the prostate. This is a very basic query, but these queries can go very far in complexity and also in power that they bring to the user. So, for example, if you are dealing with very large collections such as an LST and you have 10 terabytes of data, it's probably wasteful to download the entire collection to your computer just to find out that some of those images have missing slices or some other problems. With the interface such as this, you can select just those images that are geometrically correct, or you can select a cohort which contains a representative mix of different vendors or different patient body weight. And you can download only those images that you need. So here with SQL, you can really use metadata as a map to navigate a very complex landscape of data and files. So, and here are just some of the, just a little bit of samples of questions you can answer with DICA metadata and SQL. And we have a lot more examples in IDC documentation, if you're interested. Of course, not everyone is comfortable using SQL. It's a very low-level tool, but and to mitigate that complexity, we also have examples how to use Google Looker. And Google Looker is free, publicly available commercial tool, which can be used to build interactive dashboards. So with the dashboards, you can hide the complexity of metadata behind this relatively intuitive user interface. You can expose only the specific attributes that are important for a case. So for example, we have in this example, it's a dashboard that helps you navigate, understand what kind of contrast CT images that are available in Imaging Data Commons. So it will give you the information about the contrast agent, resolution, manufacturer, and you can build your cohort. You can view the images through the URLs that are included, and you can download images that you like. In this another example, it's a summary of a human tumor atlas collection, where you can search for the specific channels available and anatomy. You can gain better understanding of the data, and all of this metadata that powers these dashboards is extracted from the harmonized representation into DICOM. So very complex behind the scenes, but rather hopefully a lot simpler once you have this dashboard that anyone can build with minimal technical experience and perhaps help the clinical collaborators zoom into specific aspects of the data. Here's another example. And again, this example is accompanied by a link to the notebook that you can try out yourself. In this case, it's a Python notebook that allows you to understand the variability of acquisition parameters for various MR sequences available for the prostate that are available in IDC. So very quickly, you can get a summary of TRs, TEs, flip angle values, manufacturers, how they're varying across the collections. And without leaving your notebook, you can select the subsets of interest and visualize those images directly in the browser. So with this notebook, you can have a little lab where you can gain better understanding of the data. You can decide, does it meet your requirements for, I don't know, developing or evaluating certain algorithm? And all of that can be done without explicitly download anything, installing anything, signing into any system. So we hope that I really think that it's an example that highlights the power of metadata and tools available in IDC. I'm sure that some of you perhaps heard about Hugging Face. In a similar manner, if you are developing tools that you want to share with your readers, for example, or your customers in Hugging Face, instead of relying on PNGs and various other sources of data, which is quite distant from clinical practice. You can integrate your algorithm to demonstrate its functionality directly on the data available from IDC, which is somewhat more than the PNGs you may find in various repositories. So in this example, again, you have a link here. You can access this live demonstration. You can the source code is available under public license. We took an algorithm developed by others. So we did not develop this algorithm, SegVol, but we thought, okay, this is an interesting algorithm. It does volumetric segmentation. We have relevant data. And Cosmin Susu, who one of the engineers working with me, developed this demo really in a matter of a week, perhaps, without any prior knowledge about Hugging Face. So with this demo, you can select a scan from IDC, and you can choose the semantic prompt, select what you want to segment, run it directly in your browser, and see the result. So if you're writing, next time you write a paper, you could consider developing this kind of demo to very quickly give some idea to your reviewers about the robustness of your algorithm and help the users, readers of your paper reproduce and experiment with your work. A final set of tools that I want to highlight is the mhub.ai. So this project is a very active project, which is led by Dr. Hugo Ertz, and I'm a co-PI in this project as well. We are curating deep learning models applied to cancer image analysis. And here, the idea is that for all of the AI models that we ingest, they provide uniform interface for the input. They work directly on DICOM data available from IDC, and they provide uniform representation of the analysis results so that you can relatively easily run algorithms that, let's say, segment the same structure, compare the results, do nice visualizations, and then also you can incorporate those tools into batch analysis workflows. At this point, we have probably more than 20 tools, and we are working to add more. Right now, just about radiology, but we are working to add some digital pathology models as well. So I talked about data, I talked about tools, and the last component here is the compute. Again, here, it's not our role to establish or maintain compute resources. It's more about understanding what is already available, how to use existing cloud compute capabilities for the purposes of cancer imaging research, and develop demonstrations and best practices, help those users who are not familiar with the cloud understand what are the strengths and weaknesses of this emerging technology. So the first kind of entry-level recommendation, if you want to get started with the cloud, and perhaps many of you already use this, is cloud-based notebooks. So Google Colab really stormed into our field and is used everywhere. With Google Colab, you can, for free, get a cloud-provisioned virtual machine with GPU and the Python notebook interface to run the code and experiment with the analysis. So couple this with IDC that provides data, which is versatile, cloud-based, quick to download, metadata rich, and you can do some interesting things. So here's one example. We developed this notebook to demonstrate how you can experiment with MedSam. And here, if you check out this notebook linked at the bottom, you will see how you can pick random images from imaging data commons and in your browser experiment with the prompts and evaluate the results of applying MedSam to this data. Again, it's just an example. The point is to make it clear that you can take those open source tools, couple them with these cloud-based capabilities, and very easily prototype, understand their strengths, decide what you want to do with them without significant investment of effort. Next year, if the free notebooks are not sufficient to you, you can provision highly configurable virtual machines, choosing the GPU architecture, disk size, operating system, and you need to be careful when you use those because this will cost money if you do this through the commercial providers such as Google or Amazon. But at the same time, you pay only for what you use. So you can use this highly capable machine for an hour, do your experiment and then shut everything down and not pay for anything. If you want to have similar capability for free, we highly recommend National Science Foundation Access Program, which provides the mechanism where you can apply for cloud credit allocation. It's free for US academics, and instructions how to get started with this program. We have been very, very happy with the quality provided by this national program. As one example of what you can do once you have this very powerful machine in the cloud, here you see 3D slicer running in a cloud-based environment, and you access this cloud desktop through your browser. In this case, the virtual machine that is running behind the scenes somewhere in the cloud has 200 gigabytes of RAM. And it has that much memory because that's how much you need in order to load this very high resolution digitized cryosection visible human data set. So as you can see here, once you have that much memory, you can easily perform this visualization using a 3D slicer. Very hard to achieve on a regular desktop, but if you want to do this, you can allocate this machine very quickly, experiment with the data, and decide what you want to do next. And this example was prepared by Steve Piper as part of IDC work. But then kind of the last point I want to make about cloud-based computing is that it's not just about single machine analysis where you can have a notebook or user interactive interface to a desktop somewhere in the cloud. You can also batch process large amounts of data very cost efficiently and time efficiently. So in this study that we completed recently, we went back again to the NLST collection, which didn't have any annotations, and we applied total segmentator that segments around 100 atomic structures automatically in CT images. So with NLST, the amount of data is really mind-boggling. And if you think how to run total segmentator on each of those scans, and then also after that extract radiomics features for all of those segmented structures, it's a very time-consuming task. With one virtual machine, it would take about 2 years. At least was the basic approach. So with the capabilities available from the broad Terra platform, it's a cloud-based workflow execution environment. We were able to accomplish this computation in just 8 hours, and the cost is quite reasonable. As a result of performing this computation, you can read more about how we did it in this preprint. The goal is not to do this just for the sake of demonstrating you can do computation very quickly, but in order to enrich the data with information that is extracted from the images. So now that we have completed this, not only you have the CT images of chest for this collection, but you also have, for example, volume of the heart, or you have mean attenuation of the liver region wherever it is available in these images. And this opens new opportunities about how to make this data more usable and accessible to those researchers who do not necessarily have the expertise to do segmentation and feature extraction, or who just do not have the resources or time to work on this. So here's just a quick highlight of what you can do with the data. In this case, this is the work that was done by Tipa Krishnaswamy, instructor in my lab. And we took a study that looked at the variation of vertebral body volume across vertebra and between male and female. And you can see the published data in these two green and red lines. And this analysis was done using manual segmentation for just 40 patients. We did a similar summary for the vertebral segmentations performed by total segmentator for 20,000 patients. And we can see that the trends are different. The definition of the vertebral body here is not the same as here because we also include the entire vertebra, not just the vertebral body. So the absolute values are not meaningful, but the trends are very consistent. And this is just to highlight what you can do. You can think about similar studies looking at, I don't know, characteristics of the kidney or lung across this very large cohort and correlating those image-based phenotype with clinical attributes of this population for NLST. So I think the opportunities that this opens are quite amazing. So this is my last slide. I hope that this was interesting. We offer you a lot of data, but not just the data, but also tools that help you use the data. And we hope that by experimenting with the data available from IDC and the tools, you learn more about the cloud, about the value of metadata, and about various AI tools that are emerging these days. So we hope to have you as part of our users. Please ask any questions in our forum. We have a publicly available forum. Go to our portal. We have the documentation page how to get started. And we have some publications you will find also in our documentation. I want to acknowledge the contribution of the team, of course. And as promised, here is the link to the slides. So you're welcome to use them. Thank you. Sorry, I didn't leave a lot of time for questions. I apologize.
No, fantastic, fantastic. Boy, that's a boatload of information and data wrangling on steroids, really, and wonderful tools. Quick question: do you have a know offhand when your workshop is at RSNA? If people are going and want to attend, do you know what day that is? If not, they can look
I
it
think
up.
it's Monday, but let me
Monday.
check.
All right, it's a great hands-on workshop,
Yes,
and you
it's
can do
Monday.
that Monday. Fantastic. And attend that. How and wonderful to hear that you have really no restrictions and requirements in terms of user agreements, which often other sites do. How do people contribute to data commons? And a sort of corollary question, the de-identification of data, what sorts of tool are you doing that? Do you have tools that can help people de-identify data for contribution to the imaging data commons?
Yes. So yeah, this probably was a point that was lost in this too much information slides. But we do not have the capabilities to de-identify the data. We either defer to the entities that collect the data to de-identify, or we refer people to TCIA if those are individual submitters. So yeah.
Great. Okay, that's fantastic. And we have a couple of questions here. Is MHub similar or the same as ModelHub.ai?
So
Is that
MHub.ai supersedes ModelHub.ai. You can think about it as a next generation of the Model Hub project.
okay? We have a comment here from Andrey and Dalka. Fantastic talk, Andrey. What awesome resources. And we have another question here. Harmonizing clinical data from the IDC to the clinical schema of GDC. And thanks for the great presentation.
Yes. So there are a lot of this topic is a lot bigger than imaging data commons. Imaging data commons is about harmonizing images. We are very interested to see clinical data harmonized, but it's a lot more work than we can do ourselves. Within the broader cancer research data commons, there is a lot of discussions how to proceed with this. There are some emergent projects that may attempt this kind of harmonization, but there is no immediate timeline or specifics when this may happen, unfortunately.
Great. Well, Andrey, fantastic talk, fantastic work, wonderful tools that I think many people can take advantage of, and incredible amount of data and diverse and heterogeneous data, and multimodal, which is really quite unique. So thank you very much for doing this talk. Really appreciate your time, and thanks everyone for joining today. Fantastic, Andrey.
Thank you so much for inviting me, and thank you for the audience. Appreciate it.